In the last decade, biological research hit a critical volume threshold. Laboratories around the world are generating massive, multiplying troves of genomic data every single day, sequencing human DNA at a speed and scale that outpaces our ability to process it all. Buried within those vast digital archives are the genetic mutations that drive complex cancers and the hidden molecular anomalies responsible for rare, undiagnosed diseases. The challenge for modern biology has shifted. With sequencing now routine, the focus has moved to deciphering the millions of signals within those sequences. Having the complete chemical blueprint of human life sits dormant on a server if researchers lack the tools to navigate its contents. Look at this raw genomic output. It is a continuous, overwhelming stream of billions of A's, T's, C's, and G's. The human brain cannot extract patterns or meaning from this abstract ocean of text. To sort through the noise, researchers deploy machine learning algorithms, but these predictive models often deliver their results as a closed loop. They identify a target, but the math used to find it remains obscured from the scientists who need to verify it. If a researcher cannot visually confirm why an algorithm flagged a specific sequence, they cannot confidently act on it. The data remains detached from human cognition. To solve this, the Broad Institute developed the Integrative Genomics Viewer, or IGV. It serves as a translation engine for modern biomedical research. This software organizes the information. IGV takes that massive wall of raw code and structures it into a clear, navigable track. By aligning the text into a spatial layout, researchers can visually scan the architecture of the DNA and isolate the exact, granular anomalies driving diseases like cancer. The software scales to match newer technologies, like long-read sequencing. Researchers can zoom out to view massive, continuous stretches of DNA, revealing large-scale structural variations, entire blocks of genetic code rearranging in ways that older, fragmented methods missed. By matching the actual shape and scale of the sequences, the software converts abstract statistical points into physical targets that researchers can inspect. Biology is rarely contained to a single layer of DNA. To accurately map biological systems, scientists must integrate genomics with transcriptomics and proteomic data, an approach known as multi-omics. This diagram illustrates how IGV handles that compounding complexity. It allows researchers to stack distinct datasets on top of one another. By dropping vertical markers through these varied tracks, scientists can see exactly where different molecular layers intersect and influence each other. This vertical mapping extends to the resolution of single-cell sequencing, allowing researchers to track unique cellular diversity within deeply integrated biological networks. It provides the spatial map required to untangle the overlapping interactions between these multifaceted biological systems. As artificial intelligence takes on a larger role in parsing these vast biological datasets, the need for human verification increases. When an algorithmic wave sweeps through millions of data points to flag a hidden geometric pattern, researchers turn to IGV to recreate that finding visually. They use the viewer to physically observe the machine's logic and confirm the biological mechanism on screen. Human observation remains the ultimate check for machine logic, grounding unexplainable predictions in verified, understandable science. These laboratory tools move into clinical environments to connect raw genomic sequences to the specifics of patient care. Clinicians rely on this visual context to decode the genetic variables behind enigmatic rare diseases and to identify specific receptor targets for drug discovery. By reading a patient's individual genetic profile visually, doctors can pinpoint vulnerabilities in a tumor and customize a treatment plan, the core mechanism of precision medicine. Because medical professionals can clearly observe the underlying biological mechanics, they are equipped to make precise, informed clinical decisions. Direct visual ref. These discoveries are shared globally. Through IGV reports, scientists package their findings into interactive digital dashboards, sharing navigable genomic maps with research teams across the globe. As biomedical engineering advances, these visualization techniques are already evolving to incorporate spatial virtual reality and increasingly sophisticated algorithmic models. In an environment saturated with massive data and machine learning, visual translation provides the clarity required to turn abstract numbers into biological discovery. Translating abstract data into observable reality is what ultimately turns information into medicine.